Hello, I am delighted to welcome you today to the Drugs Payload Quantification in LNPs with New Scatter-Free Absorption Technology webinar. My name is Alistair Elmer, Director at Handsome Wade Intelligence, and I have the pleasure of chairing this meeting today. A quick note on the agenda, we're going to be hearing from 3 expert speakers. Kicking things off will be Darren Andrews. Darren is the Chief Commercial Officer at Marama Labs, and he'll be providing a brief introduction to their new CloudSpec instrument and technology. Then we'll be handing over to Dr. Johanna Simon, Head of Formulation Discovery and Analytical Characterization at Merck. She will be giving one example today of how the quantification of payloads in LNP formulations has benefited her team. And then we'll be handing over to Dr. Emily Young. She is the Head of Non-Viral Delivery at 4Base Bio, and she'll be showing yet another application case for the CloudSpec instrument and the advantages it's brought to her team as well. Finally, there will be some time at the end for Q&A, so please do feel free to put your questions into the chat, and we will come back to them at the end at this time. If we do run out of time to ask your questions today, then please do follow up with us, and someone within the teams will be able to answer your questions for you. Without further ado, I shall be handing over to Darren.
Hello, and welcome to Marama Labs webinar on CloudSpec and scatter-free absorption spectroscopy. My name is Darren Andrews, and I'm going to be explaining how the technology works in advance of our presenters, who will talk about how they're using it in their laboratory workflows. So for quantifying RNA in LNPs, for example, or other drug payloads, the thing that you have to do with most techniques, if not all techniques, is remove that RNA from the particle before you can analyze it. And that causes all sorts of problems, difficulties in removing the payload itself, difficulties with the assays, particularly something like ribogreen, which may be a bit of a challenge to get working with the kind of precision that you're looking for. It would be much easier if you could just use regular UV-Vis-like approach, where in a buffer, for example, you can measure the concentration of your payload really easily, really simply, and get an answer for your concentration that way. What CloudSpec brings to the table is the ability to measure payloads in that kind of simple, easy to use UV-Vis way, as if the particles were not scattering light, which is the thing that gets in the way of your accurate measurement. And that's what CloudSpec does. So regular UV-Vis works by measuring the loss of light transmitted through a sample by absorption. And you can turn that absorption into a concentration. We all know it, we've all seen it, we probably all do it every day. If you've got nanoparticles in there, the problem with those is that they scatter the light, particularly in the UV, very effectively. And so when you transmit light through the sample, some of it reaches the detector by absorption, but some of that is lost to scatter. And that scatter signal is not collected by the detector, and that imposes this kind of underlying scatter curve on your signal, as we see in the spectrum on the top right. And what that means is that you're not able to quantify the amount of payload in your sample, and you get the wrong answer. You could get around that if you got around the problem of not collecting all of the light because it gets scattered. You can do that if you had an impossible detector, which was a sphere that surrounded your entire sample. So all light that went in every single direction, if that was collected, then that will be able to measure the absorption of your sample. But of course, that's not really a practical solution. What CloudSpec does is it approximates that by placing the sample inside a highly reflective integrating sphere. And we don't shine the light directly through to the detector, we shine it in a different path. And that path then means that any light that's collected by the detector has to have scattered through the sample before it gets measured. And in that way, we approximate this perfect spherical detector, and we allow an easy measure of the entire volume of that sample. So whatever payload is in there, whether it's encapsulated or not, we measure that in one very quick measurement without you having to do anything, without you having to do much preparation, and without you having to know anything about the particle system itself. And where we get to with that is, if you see the top right, this is a scatter-free absorption spectrum. It's as if the scatter wasn't there. The empty LNP is there just for reference to show that there's no absorption underneath the RNA peak in this particular case. And if you compare that with regular UV-Vis, you can see that it now looks much more like you would expect an RNA peak to look. And you can analyze it in the same way that you would with regular UV vis. In terms of the workflow, well, we measure the buffer, and then we dilute the sample into a cuvette, and we measure that. The measurements take about 15 seconds each. The whole process takes a couple of minutes. This is a real example here. So we've got an SM102 type formulation. The SM102, the cholesterol absorbs a little bit below 260 nanometers, but in this case, we just measure the 260 peak and we can convert it into a concentration just like you would with other UV-Vis techniques without having to know anything about the particles themselves. Really, really easy. This is data taken from a paper that we did in collaboration with CureVac. Very much suggest that you go and read this. And here we're exploring the impact of scatter on the traditional UV-Vis signal and how scatter-free absorption then removes that and allows you to do a concentration measurement really easily and shows the linearity and precision of the measurement. So thoroughly recommend having a look at that. Finally, there's one other thing that you might want to do with nanoparticles. If you have an ionizable lipid in this case, or other lipid component which absorbs in the same region as the RNA, you have to be able to remove that signal. And actually, because scatter-free absorption is just like regular UV-Vis, but without the scatter, we're able to bring to bear the analytical techniques you would do for that. So just like when you have a composition measurement in UV-Vis and you're trying to measure, say, RNA and DNA at the same time, what you're doing there is using those two spectra and fitting them to your data. We do this here, and we can get the RNA concentration in the software without needing to do any kind of separation and working out the concentration in the presence of absorbing species. The most obvious comparison, I guess, is with ribogreen. What we're removing here is lots of steps, lots of equipment, and we're making that really, really quick and easy to use. And that's really the benefit of CloudSpec. Well, thank you for listening to that introduction. I hope it explains enough to be able to understand what your main speakers are getting from the instrument. There's some further reading that you can get if you want to take a look at our website and various other things. Thank you for attending. I'm sure you'll enjoy the two speakers who we've got lined up for you. Thank you for your attention.
A very warm welcome from my side. My name is Johanna Simon, and I'm part of the so-called Early Formulation Screening Service team, and we are developing customized lipid nanoparticle formulations for the delivery of nucleic acid payloads. In my presentation today, I would like to get started by giving you a short introduction into the RNA field and where we as a company see the huge potential of this novel modality. I would like to further highlight where we as the Early Formulation Screening Service team are supporting the development of novel RNA-based medications. And this will lead me very nicely to my two case studies, one focusing on RNA quantification using the CloudSpec technology, and the second one on focusing on protein quantification of so-called antibody functionalized lipid nanoparticles. So let's get started. I think we have all witnessed through the COVID pandemic the huge potential of mRNA-based technology. For me personally, and a lot of others in the field, this was really a breakthrough of the technology. mRNA-based therapeutics and vaccines can not only be used to prevent diseases, but also to treat them. And there are multiple possible applications for RNA-based medications. And I personally think there are two very exciting areas. And this is on one side the gene editing application, and on the other side the personalized medicine. In the field of gene editing application, we can actually repair individual gene defects and treat very severe diseases like the Huntington disease, for example. In the field of personalized medicine, I think it's very interesting, as we have seen when we studied different cancer types, that cancer is very much patient-specific. And I think that RNA-based medications could really be a game changer for personalized medications. For any RNA-based medication, you actually need a suitable delivery system. And the most advanced delivery system that has been developed and was also used in the COVID vaccines are the so-called lipid nanoparticles. The lipid nanoparticles enable the delivery of RNA into the cell. because mRNA, for example, is very prone to degradation and would not be able to reach the desired cell without a suitable delivery system. We are working with the classical 4-component lipid nanoparticle systems, which comprise of the so-called ionizable lipid, some helper lipids, cholesterol, and a PEG lipid. All of those 4 lipids have a very important function, and also the ratio of the individual lipids within a particular formulation is actually quite crucial for the successful application of an LNP. The most important compound in that LNP is the so-called ionizable lipid, as it really drives the success of a certain formulation, is responsible for biodistribution of the LNPs, for the potency and safety. As we have seen that there are multiple different applications for RNA-based medication, you might ask, why don't we have already more RNA-based medications on the market? And the answer to that is quite short. It's really hard to develop a successful RNA-based medication. On one side, you need to develop a suitable delivery system that is potent and safe. You need to have robust manufacturing processes for the RNA and for the lipid nanoparticles. And there's also the aspect of stability for the formulation, as we have seen, for example, for the COVID vaccines. You need to store them at ultra low temperatures most of the time. Bringing all of this together, it indicates that developing a successful RNA-based medication is not straightforward. We as a company want to support the development of RNA-based medications, and we have experience in developing RNA-based vaccines and therapeutics. We can manufacture mRNA in small scale for research purposes, and we can also manufacture it in larger scale for GMP. The lipids that are needed within the lipid nanoparticle formulation are manufactured within our company for more than 25 years. We can also do that in a small scale, but we can also have custom lipid synthesis for individual compounds within a formulation, also under GMP. Bringing together now the lipids and the RNA is exactly what we in the early formulation screening service team are doing. We are developing then the specific lipid nanoparticle formulation for a certain application. And to end that, we have a team located in the US that can manufacture lipid nanoparticle formulations in a GMP setting. So all of that together, we as a company really want to support you from start to end to enable the development of novel RNA-based medications. Next, I would like to focus a little bit more on where our focus is. So we operate completely in the preclinical space. And as I initially described, it's our ambition to design customized lipid nanoparticles for a certain application. So how are we doing that? As I described in my introduction, the ionizable lipid is really the main, the most important compound within the LNP formulation. So we have access to libraries of novel ionizable lipids. And those novel ionizable lipids have been tested already for a variety of different applications and already in very different client projects. So we would basically start off by doing a so-called formulation screening, where we would actually test different novel ionizable lipids. Once we have identified that a suitable ionizable lipid, we would then do some formulation optimization to improve the properties of the LNP. We also are looking at a stabilization of LNPs, so we can either do that for liquid and frozen LNPs, but we also have capabilities to freeze dry LNPs to enable storage at higher temperatures. Last and very crucial is the analytical characterization of the LNP. LNP formulations, as you have seen, are very complex formulations, so you need to have suitable analytical tools in hand to characterize that formulation to ensure a high quality of your product. And this is where we also have put a strong emphasis on to have a broad variety of different analytical tools for characterizing LNPs. And that's why it fitted very well that we got the opportunity to test the CloudSpec instruments in our lab to investigate if that's a suitable technology that we can add to our analytical portfolio. As the RNA field is evolving, there are now both on the European and the US side, there are both guidelines describing the critical quality attributes of LNPs. I would like to mention to highlight especially two different critical quality attributes for the LNPs. This is on one side the LNP size and polydispersity, because the size and size distribution of a formulation is really important as it affects the stability, encapsulation efficiency, cellular uptake, toxicity, biodistribution. So it's essential to characterize and control the LNP size. The second very important critical quality attribute is the RNA content, because the RNA content within a LNP formulation is actually the value that is also then later needed to precisely dose the RNA-based medication. So therefore, you need reliable assays to quantify the RNA content within the LNP formulation. And this is exactly what we wanted to test using the CloudSpec technology. Starting off with the first case study. So the classical assays that are used for RNA content determination are the so-called ribocrene assay. Shown on the left side, you can see for that classical approaches, you basically take the LNP, you add a detergent to disrupt the LNP, and then the RNA is released. The RNA later on can then be quantified using some fluorescent dyes, such as the ribocrene dye, or you can also do some UV-Vis measurements to measure the RNA content at 260 nanometer. In case of the absorbent-based measurements, of course, you need to use detergents that are UV compatible, that you don't have an interference in your UV-Vis spectra. All of those classical assays, they have all in common that you require sample preparation. So you need to break up the LNP to release the RNA and quantify it. For the CloudSpec technology, you actually do not need any sample preparation. So what you actually do for that measurement is you take the LNP, you dilute it, and then you simply measure, put the cuvette in your instrument, and you measure the RNA concentration. So this so in our first case study, we wanted to evaluate the CloudSpec technology and compare it to also the classical approaches to see if there are any differences in RNA content. We further wanted to also evaluate the suitable concentration range where we can work with in the CloudSpec instrument. And as I also described, as we typically have plenty of different formulation with different ionizable lipids or different RNA types, different compositions, of course, we wanted to evaluate if there are any impacts coming from the formulation itself or for different RNA types. So starting off with our first set of experiments, we manufactured very classical LNPs containing mRNA, very similar like the COVID vaccines. And we then determined the RNA content within that LNPs using 3 different methods: the ribocrene assay, our UV-Vis content assay, and the CloudSpec technology. And the values you can see in the table shown above. And when you compare the individual values for the RNA content between the different assays, you see that there is really a minor deviation between ribocrene assay and CloudSpec, and also UV-Vis content and CloudSpec. So this was the first set of data that we generated, where we saw that the CloudSpec technology really gives reliable, good data for RNA content quantification. We then further did a series dilution of the LNPs at different concentrations, going from very low concentration of 2 microgram per milliliter to a higher 20 microgram per milliliter. And as you can see in the plot, we have a very nice linear regression curve. So you can even determine the RNA content at the very low range of 2 microgram per milliliter. this was also very interesting for us because, as I mentioned, the other very important critical quality attribute is the size determination. And for size determination using dynamic light scattering, we also dilute the LNP roughly to a concentration of 2 microgram per milliliter RNA. And so that showed us that we can actually use the same dilution for the measurement in our DLS instrument and for using the CloudSpec instrument. Then we further tested a variety of different formulations. Today only shown here some examples. So we on one side manufactured different mRNA-loaded LNPs with different ionizable lipids within the formulation. And also we manufactured some siRNA formulations, also here again with different formulation compositions. We in both cases did a serial dilution of the RNA to then also determine the exact extinction coefficient, and this extinction coefficient was then also used for the calculation of the RNA content. We compared then the RNA content determined by ribogreen, UV-Vis, or by the CloudSpec instrument, and you can see in both cases that the deviation for the formulation between those assays is very low. So in both cases, for mRNA-loaded and siRNA-loaded LNPs, we could successfully quantify the RNA content using the CloudSpec technology. So this was very encouraging data for us, and it really demonstrated that the CloudSpec instrument is suitable for a variety of different LNP formulations and is a reliable method to actually quantify RNA content within LNPs. Now coming to the second case study that I wanted to share today with you. In this case, we tested so-called antibody functionalized LNPs. The antibody functionalized LNPs are a novel formulation, and where you basically have a targeting ligand on the LNP surface. And this targeting ligand should enable an even more specific cell interaction. Of course, with adding a certain targeting ligand towards the formulation, you add another complexity. And of course, what you also need to achieve is then quantification of this targeting ligand on the LNP surface. So in that second case study, we wanted to investigate on one side if the antibody on the LNP surface is having an impact on the RNA quantification. And in the second set of experiments, we also wanted to quantify then the antibody within that formulation. Because the typical assays that are used in the field for quantification of antibodies are typical protein quantification assays, such as the BCA assay, or you can also do some UV-Vis-based measurements at 280 nanometer. But what we have seen in the past is that both of those assays show a strong interference with the LNP matrix, and a lot of times you do not get reliable data for your protein quantification in the presence of the LNP. So we started off by doing the first set of experiments, and in these experiments we basically took the LNP formulation and then we added antibody within that formulation. We then quantified at 260 nanometer the RNA content, and we compared basically, here shown in blue is the RNA content of the pure LNP, and shown in pink is the RNA content of the LNPs in the presence of the antibody. What we could nicely show is when you compare the blue and pink graph is that there is really a minor deviation between the RNA content determined in both settings. So this really indicated that the antibody within the formulation does not have an impact on RNA quantification, and this was really an encouraging result we got. Second, we also, as I described, we wanted to quantify the antibody itself within the formulation. So for that set of experiment, we first diluted the antibody to different concentrations to determine the protein concentration. So we used also there the concentration range between 12.5 microgram down to 1.5 microgram per milliliter, shown in green. And then we added again the same concentration of antibody to the LNP and measured then the antibody concentration at 280 nanometer, because this is the absorbance maximum for most of the proteins. What we saw when we did the measurements and investigated the spectra at 280 nanometer, we saw, shown on the graph on the right, we saw that the LNP spectra itself had also an interference. So there was quite a huge signal from the LNP at 280 nanometer. Therefore, we decided to subtract basically the LNP spectra from the naked LNP, from the LNP spectra from the LNP in the presence of the antibody. And by doing this, we could then quantify the antibody. When you compare now the absolute values, so the graph shown above, so the values for the protein in the presence of the LNP shown in yellow, and then the protein concentration, the pure protein concentration in the absence of the LNP shown in green, and you compare both of those values, and you see that there's also a very minor deviation, especially in the lower concentration range between 3 to 6 microgram per milliliter. And this was a really nice data we obtained, as we really could show that we can quantify the protein in the presence of the LNP. Of course, when we do that subtraction, but we could reliably quantify the protein then in the presence of the LNP. And this method could really be used for characterizing those complex formulations containing antibodies. With that, I'm at the end of my presentation. I hope you enjoyed having a look at our case studies and getting some idea about the CloudSpec technology. I'm very happy to get in touch with you to give you more insight, and looking forward to your questions now. Thanks a lot.
Hi, I'm Emily. I'm the head of non-viral delivery at Forbis Bio. I manage our in-house lipid nanoparticle platform called Hermes, and I'll be talking about our experience of implementing the CloudSpec for RNA and DNA quantification in our LNP workflow. So a little bit about Forbis Bio. We're a synthetic DNA company. We manufacture DNA entirely enzymatically as an alternative to plasmid DNA for use in various different gene and cell therapy applications, including viral vector production, DNA vaccines. We have IVT templates for mRNA therapeutics, and also specific molecules for gene editing as well. But all of those molecules we also encapsulate into our Hermes non-viral delivery system, and we have specific nanoparticles optimized for all of those different DNA and mRNA payloads. So just to give you an idea of the molecules that I'll be talking about in terms of nanoparticle encapsulation, we have a few different modalities of DNA at Forbis Bio. All are linear molecules produced enzymatically, but we have our IVT template, or opDNA, which has a 3' open end, which means we can feed directly into the IVT reaction without need for enzymatic linearization like you would with plasmid DNA. We have a hairpin DNA, which has its hairpins at both ends, making it very stable. This is typically used for viral vector production, and also is what we encapsulate in our nanoparticles for DNA vaccine applications and any gene replacement applications as well. Then we have our open-ended DNA, which has a stabilized open end on both ends, and we've recently started enzymatically producing single-stranded DNA as well. Both of these are used for HDR templates. The single-stranded DNA has advantages in terms of immunogenicity, and by producing it enzymatically rather than chemically, it means that we are able to make much larger amounts and also much longer sequences compared to other methods for single-stranded DNA. So just to very quickly take you through our DNA synthesis process. We're not a de novo synthesis company, so we do require DNA in some format before we start our amplification. This can typically be from a plasmid if one is already available, but we can also work from PCR fragments or DNA G blocks as well. We generate a circular backbone-free DNA template, so that doesn't have any of those antibiotic resistance genes or any bacterial sequences like plasmid DNA. We are never amplifying that during our process. We go through a cell-free DNA amplification process, entirely enzymatic, which is then taken through to our DNA processing, where we add those stabilized or hairpin ends to the DNA, which then goes through to a very nice DNA purification process to remove all of those enzymes that we used during the synthesis, and then, of course, onto QC. So to position synthetic DNA against plasmid DNA is extremely cost effective. We're talking about making DNA in a bag rather than a large scale bioreactor, which has low labor and capital demands compared to plasmid DNA. We also have our own in-house enzymes, which allows for quality control and reduced costs. We have very flexible scale, so at GMP we're currently operating between 50 mg and 10 grams, and at RUO grade we are offering from 1 mg. In terms of safety considerations, the lack of bacterial backbone in our final product and during amplification obviously has regulatory considerations, but because we're not using cells during this process, it means that we don't have to worry about endotoxin contamination, so we have low bioburden, and we also have a lack of, we don't have to worry about bacterial genomic DNA in the process. Really, the advantage is speed, particularly at GMP grade. With plasmid DNA, you're looking at lead times of 6 to 9 months. We can do that in a matter of weeks with synthetic DNA. Also importantly, we have good sequence flexibility compared to E. coli. So we are suitable for complex sequences. So for example, those high GC contents, IDRs, for poly A tails, we can also encode up to 120 base pairs into the DNA template. Which is an advantage, and we don't have to worry about the homologous recombination that occurs in E. coli. We are actually suitable up to 20 kb sequences now, and on the lower end, we can synthesize as small as 100, 200 base pairs, and also it's suitable for sequences that might be toxic to bacteria. So Hermes is our internal non-viral delivery platform. We use all of the conventional lipids of an LNP, but where this platform differs is that we also add a cationic scaffold to the nanoparticle, to which we can add other modalities such as a targeting ligand depending on the application, or we can use the cationic scaffold in isolation for stability purposes. We've really optimized this method so that we add that fifth component in a one-step microfluidics method. So we repurpose conventional microfluidics setups to add this into the dilution channel, which means that it's mixed in one step and there's no need to add that ligand with conjugation methods post-assembly. We've optimized formulations for all of our payloads. So of course we deliver mRNA, but we've also optimized specifically for DNA and also for our different gene editing payloads as well. We've really started to implement the CloudSpec in our normal day-to-day workflow of nanoparticle preparation. So initially, we take a naked payload reading with the CloudSpec and one other method, for example, nanodrop or an Elvis plate, to confirm that they are comparable. We use that concentration in our calculations for the nanoparticle preparation. And then, of course, we assemble with microfluidics and take a reading with the CloudSpec post-encapsulation of the LNP. We then take that nanoparticle through to normal downstream processing steps, for example, dialysis in the neutral buffer, after which, again, we'll take another reading. And then, of course, if we do any other processing, such as TFF or sterile filtration, we'll take another measurement here so we can get an idea of yield of the nanoparticle. So, of course, the first thing we wanted to know was whether the CloudSpec could measure naked payload concentration compared to some very standard methods, so nanodrop or the Clarios or Elvis plate. So we screen various different nucleic acids from our synthetic DNA, plasmid DNA, or mRNA, and you can see that the CloudSpec gives very similar naked payload readings compared to each of those, and it's very accurate and reliable. So to give an example of what the CloudSpec can do in terms of lipid nanoparticles and how those measurements look throughout the process, these are just some nanoparticles. The formulations are not really relevant, but they are encapsulating mRNA in this case. The nominal concentration is just what we have calculated from the naked payload concentration that we took prior to encapsulation and any volumetric changes that occur during those processes. And in this case, the nanoparticles were measured pre-dialysis and then just a standard post-dialysis measurement in a standard dialysis cartridge. And you can see that pre-dialysis, all of those nanoparticles measured with the CloudSpec give very similar readings to those of the nominal concentration. Of course, the nominal relies on accurate volume measurement with the pipette, so it's not entirely accurate. So you might not expect it to be exactly the same, but we are within 5% of all of these formulations with the CloudSpec, so I think it's fairly reliable. After dialysis, in this case, the concentration was expected to go up very slightly as per the nominal concentration, but that is measured with a pipette, not particularly accurately. And you can see that the CloudSpec was able to detect a slight increase in concentration compared to the pre-dialysis measurement for all of those formulations, although again it's still slightly below the predicted, but we are not expecting 100% yield. You're never going to take all of the volume out of your dialysis cartridge, so wouldn't expect it to be entirely 100% yield. Now, compared to the ribagreen, you can see in this particular experiment, the ribagreen was measuring really low. This is the standard assay in the field, and it's always the elephant in the room. No one really likes the ribagreen assay, and this is the reason why, and that's why I've left it in. I think in reality, if I hadn't had the CloudSpec in this particular experiment, we would have repeated that ribagreen assay to make sure we had an accurate reading, because it's so much lower than what we're expecting. And when this is about 25 to 45% lower than the expected concentration. But I'm going to leave that there for now and come back to that ribagreen observation later. Also, part of our normal workflow might be to TFF concentrate the nanoparticle. So to give you an idea of what that looks like on the CloudSpec, here again you can see the nominal concentration. In this case, we are encapsulating HPDNA, so we're using a picogreen rather than a ribagreen assay, which is a very similar reagent, but it binds to double-stranded DNA rather than rather than mRNA, which is what ribagreen is optimized for. And you can see, compared to the CloudSpec, compared to the nominal concentration and the picogreen, the CloudSpec gives very similar readings. And when we concentrate, the CloudSpec is absolutely able to detect that increase. A little bit of discrepancy compared to the picogreen in this occasion. Compared to the nominal, it is lower, but that is expected with TFF. We're not expecting 100% yield. And that's what we see also with the ribo and picogreen assays. And on the right-hand side here, you can also see a cholesterol assay for this particular formulation. So that's another method for detecting the cholesterol within the formulation. And I've just presented that as the fold increase in concentration of the formulation. Obviously, they're different components. But you can see that fold increase with the cholesterol assay correlates very well with the CloudSpec, and in this case also the picogreen. We've also found the CloudSpec really useful for when we have combined payloads. So we often are looking at different types of DNA modality, but we also encapsulate those in combination with a Cas9 RNA and a guide RNA. Now, there's different reagents depending on the nucleic acid that you use. So ribagreen is really optimized for mRNA compared to picogreen, which we prefer for double-stranded DNA. And for single-stranded RNA, you also have an oligreen reagent as well. In fact, the single-stranded DNA doesn't work for things like the picogreen at all. And that really complicates it when you're screening multiple different combinations or different formulations in parallel, and you start getting into questions about which one of those assays is optimal, and also thinking about different standard curves for your combined payloads. With the CloudSpec, you don't need to do that at all. You can get a combined concentration reading, which you can see here for our combined payloads. It's very similar to what we expect the nominal total concentration to be. They all absorb at the same wavelength, so you can get a combined result very easily compared to with those typical ribagreen assays. Now, I just want to talk a bit more about the ribogreen assay. I've never spoken to anyone that particularly likes the ribogreen assay, but we, with the Hermes platform, have our own specific issues with it when using it for total concentration. So what we show here is nanoparticles encapsulating an mRNA payload with either no ligand, a low amount of that Hermes ligand, or a high amount of those Hermes ligands. And one of the problems we've always experienced with the ribogreen assay is that at high ligand concentrations, we're not often able to fully solubilize the nanoparticle. It's a cationic molecule and it really drives that payload concentration. So we just find it really hard to find a surfactant that breaks those nanoparticles. So here we've used different surfactants, Triton X at a low and high concentration as is standard. We've also used SDS at low and high percentage and a tween 20 at very low and a slightly higher percentage. And now what you can see is the naked DNA. This is just a normal agarose gel. You get this population here. For untreated, as in no surfactant formulations, you will see in every case you just get the payload retained in the well. It's not able to migrate through the well at all. When you have complete solubilization of the nanoparticle, you get a nice band of similar length to your naked payload without any smear. This is what I would consider naked, complete solubilization, and therefore you're able to measure your ribogreen, your concentration with the ribogreen, because you've released all of that mRNA from the nanoparticle. What happens when you get incomplete solubilization, which is the case with this low percent of SDS, is that you start to see this smear. So some of that payload has been released, but not all of it. And then when you don't get any solubilization at all, it's entirely retained in the well. So you can't measure the payload concentration at all in this context. Now, where this becomes tricky for us and our particular formulations is that those standard reagents used to solubilize nanoparticles, Triton X and tween, do not fully solubilize our Hermes nanoparticles at high concentration. So you can see at no ligand concentration, your high 1% standard, the Triton X, fully releases. At low ligand, we also get full release. At high ligand, you can see here that that Triton X is not fully releasing the payload, nor is a high percentage of 220. Now we do find that 1% SDS is fully able to release that payload, although it's not the best gel. However, 1% SDS interferes with the ribogreen assay, so we can't use that. At 0.25% with the high ligand, it actually works a little bit better, but you can see there is still some smear. So this has always posed an issue for us with our specific formulations, and we can never truly trust which formulations are going to lies without actually running a gel or seeing strange results and then having to retrospectively confirm what was happening, which on a day-to-day R&D when you're screening lots of formulations is really complicated. So just to show what that looks like for these exact same formulations in terms of the ribogreen assay, you can see for those formulations that didn't lyse at all, so the low surfactant concentrations in here, you basically don't measure any payload concentration because you haven't released the payload at all. When you get what I would consider full release of payload, so in this case the Triton X and the tween, which is this one, you can see that you get a much higher concentration, although it is still typically a little bit lower than what we've been measuring with the CloudSpec. And so in this case, about 15 and 9% difference between that Triton X at 1% and the CloudSpec measurement. I do wonder whether that difference, because it is consistently lower, the ribogreen than the CloudSpec, so I do wonder whether there is still some mRNA that's bound to other components, which is impacting that measurement. But what you can see with the high ligand formulation, that the difference between the CloudSpec and the high ligand is even greater, 30%. So I think with these high ligand formulations, actually, we are not getting an accurate ribogreen concentration ever with any of our surfactants. because we're not releasing that payload. But what the CloudSpec has allowed us to do is to just remove that surfactant step entirely from our process. So we're just not needing to use those anymore because we're getting that total payload concentration from the CloudSpec, which is so much simpler and removes all of that doubt and the variability between formulations. And I think we're not the only ones that experience this kind of problem. So I think you've probably all seen this paper that came out at the end of last year about the ribagreen assay and how we use it. And I just wanted to show some formulation differences in this paper to show my point. So here we are looking at the total mRNA quantified as a percentage of ribagreen to HPLC. So what you can see is that for formulation 1, you have 100%, which means the ribagreen is measuring the same concentration compared to HPLC. And you can see there are a difference again with those surfactants. In formulation 2, however, we are looking at about 80% compared to HPLC, which to me suggests that the ribagreen is not measuring as high as the HPLC. So I think here there is again a formulation difference, meaning that the surfactants are not fully lysing this particular formulation. And the difference between these formulations is just an ionizable lipid. So I could think it might be SM102 versus C12200. But the difference is just the ionizable lipid, and still we are seeing those questions. And therefore, with the ribagreen, you'd be under quantifying your total payload concentration for this formulation too, which, if you're going downstream for animal experiments or cells, whatever your application is, you could be underdosing if you're trusting the ribagreen assay. In terms of encapsulation efficiency, it actually makes relatively little difference, because if you have a very low free concentration, that relative percentage does not make a difference. So, in this case, we're looking at about 96% for formulation 1, and even though it has a lower total mRNA concentration, formulation 2 is still showing an encapsulation efficiency of about 94-95%. So it really only makes a difference of 1 or 2% in terms of encapsulation. So I think for encapsulation, really the most important measurement is that free payload concentration compared to the total payload concentration. But what we've started to do now for our encapsulation is a bit of a hybrid assay to kind of solve this issue with the complete solubilization. So this is the formulations that I showed on a previous slide, where we saw really low ribagreen compared to CloudSpec. But what we do now is we use the total payload concentration quantified by the CloudSpec, and we run a ribagreen, but only on the free payload concentration, which is half the amount of work, or even less, half, because you don't need to add any other surfactant. So we take that free payload concentration from the ribagreen only, and then of course divide that again by the total payload concentration to get your encapsulation efficiency. And what you can see for that set of formulations is actually we are getting very similar results to the ribagreen, despite the fact that the ribagreen had so much lower total concentration, because we have a low free concentration as a percentage. It's much lower. You can see for all these formulations, we're about at the expected 90-95%. For these two formulations, which had a slightly lower encapsulation efficiency, both the CloudSpec and ribagreen assay could detect that drop. So it really correlates in terms of that. It's just the total concentration, I believe, that poses challenges with the ribagreen assay. So just to summarize, the ribagreen assay relies on the use of surfactant to completely solubilize the LNP. And it's really widely assumed that Triton X at 0.5 to 1% will solubilize the LNP. Well, I think I've shown that that is not always the case for different formulations, which leads to questions on the reliability, particularly for the total payload concentration. Now, that might be able to be mitigated by using different surfactants, but it's not really practical on a day-to-day basis to be screening different surfactants for your formulations and matching that different surfactant to the formulation you want. But the CloudSpec, I think, presents an alternative to that ribagreen without having to use any surfactant at all. You can use that for an accurate total payload concentration, but you can also use that in a hybrid encapsulation efficiency assay, where you just use the free payload. And having those accurate measurements is really important for downstream applications. For example, having a correct dose for your preclinical or clinical use.
Well, thank you to all our speakers for sharing their insights today. I think we can all agree that this is a really exciting piece of technology with a lot of potential. Now, as promised, we do have some time at the end for questions. We've had a few come in as the speakers have been talking, but to all those still currently listening, please do feel free to add your questions into the chat now, and we'll see if we have time to get to them. To begin with, we had a couple of clarification questions on the CloudSpec capabilities. So I'm just going to turn to Darren quickly. If you could just give a little bit of insight as to whether the technology has been tested in non-lipid nanoparticles, and what about the labeling of peptides as well?
Sure. So in terms of non-liposomal formulations, I mean, we've done things with liposomes or LNPs, but hopefully you'll see a peer-reviewed paper talking about polymeric nanoparticles with RNA embedded coming out soon. That's out for review at the moment. Basically, polymer encapsulating the RNA is even harder, or can be even harder to get the RNA out than lipid nanoparticles. And in fact, in that particular case, CloudSpec was the only really viable technology to measure the RNA inside the polymer. So it worked really well for that. That paper, I hope, will come out quite soon. If you want to hear any more details about it or try it on your system, let me know. Very happy to do that. And then in terms of other things, well, we heard about antibodies from Johanna, and if peptides is an alternate payload, there's no reason why that wouldn't work as long as it has a UV signal to quantify it with. I mean, what CloudSpec basically does is it measures UV and gets rid of the scatter. UV vis gets rid of the scatter. So if it absorbs in a way that you can quantify it, just like RNA and DNA does, then we can get rid of the scatter and you can measure it just using the UV. So yes, there's no reason why if it's not UV active, you can't measure it.
Perfect. Thank you for that answer, Darren. Now, we also had a couple of questions come in from Jesse and Ramu, just clarifications about the work that you were doing, Johanna, in particular. So could you, in your experiment, see how much RNA A versus RNA B there was in an LNP, or was it just the total? And finally, could you also tell us whether the antibody was conjugated to the LNP at all?
Yeah, thanks for that questions. For the first part, the question was for when we have two different RNA payloads. So in our case, we only had there one payload type, but in case you would have more payloads, like also Emily showed in her data, you would actually quantify the total RNA concentration. So as a sum, you would not individually quantify the individual RNAs, but you would actually the total RNA concentration would be quantified using the CloudSpec technology. And maybe for the second question, for the antibody functionalized LNPs. So in the case studies which I showed today, we basically did some proof of concept studies. So we took some LNPs and then added antibody to that formulation. So in that case, the antibody was not conjugated with a covalent bound to the LNP, but we have done further internal studies where we had actually the antibody conjugated covalently to the LNP surface. And you could use the same principle like I described today in my presentation. You need to basically subtract the spectra of the naked LNP, and then you can reliably quantify the antibody that is actually then conjugated covalently to the LNP surface.
Excellent, thank you. We have another question about the LNP, which is about the purification in particular. So perhaps this one would work well for you, Emily, if you feel able to comment on this one. Did you need to purify the LNP from the unencapsulated nucleic acid before quantification?
Nope, so it's non-destructive. You just literally measure your full encapsulated LNP in a cuvette, very much like you would for a DLS measurement, for example. So you just dilute your full nanoparticle, and pop that in the CloudSpec with a very quick 2-minute reading.
Excellent. And another question that comes through, I think this relates to what you were saying earlier, Johanna, about the being able to tell the different types of nucleic acids. So if we decorate the LNPs with an aptamer, could it quantify that separately as well? If we've got the different ones you're measuring the total, what if you have an aptamer associated with the LNP?
You mean different antibodies on the LNP surface, for example? If this was in that direction. So yeah, if you would have also different ligands on the surface, let's say different proteins on the surface of an LNP, so yeah, you would basically then also again quantify the sum of that. So yeah, if there are different ligands, let's say this would also be then a total sum for total protein concentration that you would quantify.
Thank you. And we did also have some interest in understanding about the difference between the fluorescence-based assay and the UV-vis-based assay, comparing how well they're done. Now, I know, Emily, you spoke about this a lot within your particular talk. To see if there's anything that anyone, either Darren or Johanna, would like to add to what Emily said, to any further information on that.
Yeah, maybe just to shortly add from our side, I think what Emily described, like that it really depends on the particular formulation, how well also the ribocrene assay works, is definitely something that we have also experienced in our hands. So from the data that I've shown today, so these classical formulations, let's say, I think we get quite reliable data there also with the ribocrene assay, but we have seen similar, like Emily also described, if you have maybe cationic lipids within your formulation, so then you get quite a high deviation between ribocrene assay or some UV-vis-based measurements where you also need like detergents to break up the LNP. And I think that's exactly, I can underline what Emily presented. So I think the CloudSpec technology is there more universally. So it's a simpler approach for really a variety of different formulation compositions instead of having a detergent-based assays where you always have where you always need to ensure that really the detergent breaks up fully the LNP. And this is, I can fully underline what we have seen.
I think all I would add to that is, I guess that what we're explaining here about breaking the particles up is about essentially a method development step. So for some particles, it might be very straightforward. An example that I talked to earlier, the polymeric ones, it just doesn't work. You can't get the RNA out of the polymers in that particular example. So you are effectively removing a method development step and a method qualification step that you don't really necessarily know a priori whether it works well or not well. And then for the ribocrene assay part itself, that is something that is specific to the RNA you're using, for example. So if you change RNA types, you need to really calibrate it with that RNA type for the best answers, which might be waste of RNA. You might think of it that way. So I think there's all sorts of reasons why you might not want to break the particles up, but it ultimately comes down to there's additional method steps and potential method development, method validation that you have to do to be able to rely on the results. And I guess the CloudSpec means you don't have to do that.
Wonderful. And we do have another question actually from your namesake, Darren. We have another Darren in the room. And I'm going to ask this to you because it's about the future of the CloudSpec instrument and whether there's any plans to reduce the volume of sample for measurement, so going towards a plate-based HT measurement.
I suspect that's two questions, but I'm not sure. So in terms of the volume of sample, so it essentially works off mass. So we have a range of between say 2 and 20 micrograms per ml. You can probably go a little bit lower in some cases. So your mass is fixed. The dilution is just what you're doing to get into the 1 milliliter. That's essentially what makes the system relatively easy to use because you can get the sample in and out. In terms of higher throughput, that's certainly something that we're thinking about. Bearing in mind, we only launched this product about 4 weeks ago onto the market. That feels very much like tomorrow's problem, but certainly that's something that we've got in mind. clearly what we're trying to do now is make as much value out of the product that we've just launched.
Wonderful. Well, we've come to the end of our time available today. So I want to thank all our speakers today, Darren, Johanna, and Emily, for their very interesting talks. For everyone who's listening, thank you very much for attending today as well. Please do continue to ask any questions if there's anything that comes to mind. One of the speakers would be delighted to follow up and speak with you more about it. And yeah, everyone who didn't get a chance to get their question answered today, please do shout and let us know. Otherwise, we thank you very much for your time, and I hope you enjoy the rest of your day.